Welcome to the basic genetics course. There are 3 teachers in this course, Dr. Richa Are, Dr. Madhuji Tapadia and myself. We are from Department of Zoology, Banaras Hindu University. I am a retired person. This is week 8, 5th lecture. In my last lecture, I was talking about anomalies in chromosome structure. I am continuing with this same, but the meiotic consequences in translocation heterozygotes. In previous lecture, I did talk about inversion heterozygote. Now I am talking about in translocation heterozygote. Once again, I am bringing a common picture that we are talking about chromosomes, and chromosomes are made up of DNA. That means any kind of change in chromosome actually having change in the genes. So, if suppose there is a translocation, one part of chromosome goes to another chromosome, that means some of the genes are getting transported to other chromosome and vice versa. So, this is translocation. In my one of the lecture, 3rd lecture, I did talk about translocation. In translocation, one chromosome, this is a chromosome and this is a non-homologous chromosome. We are not talking about 2 homologous chromosomes. So, if this part of this chromosome, is cut from here and goes to this chromosome, and this chromosome's part coming here, then this you call it as translocation. So, here after translocation you have the translocated, this is blue part, this is translocated from the other one, and this red part is translocated from the other one. When such kind of translocation occurs, and if the translocations are in heterozygous condition, that means out of the 2 chromosome, if I go back to the slide, out of this is one chromosome where translocation occurred, its homologue is normal. Same way the translocation occurred in this chromosome, its homologue is normal. So, now I am showing you, you can see normal, N1 is normal. Here you have the chromosome which is normal, T1 is the translocated one, where this has the normal part, this red one, and then blue part from the other non-homologous chromosome. So, this is a pair. The other one, a blue chromosome, that means this is another chromosome, and here you have the part of blue and part of red, that is the translocated chromosome. So, these 2, this one and this one, these 2 are the translocated chromosome, and this are the normal chromosome. Because this chromosome has a homologue here, so during pairing they will pair. Because this is having homology with this, during pairing this will pair with this. This is having this red portion, so this will pair with this. The blue portion, this will pair with this. So, that means this is a complex situation. In this complex situation, you can see pairing. This is red, normal one. With what this will pair? This will pair with this part. So, this is pairing with this. If it is pairing with this, the blue part will be here. This red is pairing, this much will be pairing. So, this is pairing. This much will be unpaired with this, but paired with this. This will pair with this blue one. This blue one will pair with this. So, this will be a condition that 4 chromosomes will come together during meiosis when it has to pair. In my last lecture, I said that chromosome pairing occurs at zygotine during meiosis. That means at that time, each chromosome is having 2 chromatids. So that is why here each chromosome with 2 chromatids are being shown. When this kind of pairing occurred, this is the pairing occurred. Then after that there will be segregation also. If the pairing is between the 2 homologous chromosomes, the 2 homologous chromosomes will tend to separate, and in the process, if there is a chiasmata formation, a part of this, a part of the other chromosome will remain together. Here, if you see how it can become separate, there can be one way of separation, that this and this will remain together. When we are drawing here in a 2-dimension, you are seeing this, that this paired with this paired with this paired with this paired with this. But if you see in 3-dimension, this kind of pairing can be like this also. So if the situation is not planar like this, that means this and this can go together, this and this can go together. So this is one kind of separation, that this has gone together and this has gone together. So you can see this red, this one and this blue. This has gone together to one pole, and this one and this one has gone to another pole. Because these are the 4 chromosomes were together. So in one pole, the 2 chromosomes will go, another pole, the 2 chromosomes will go. The other kind of possibility, that this, let us say this goes to one pole, this goes to one pole, this goes to another pole, this goes to another pole. This is the situation. This red one has gone there, this one has gone to the one pole, and this side, there. The third possibility is, because this is in 3-dimension, all the possibilities are there. These 2 can go to these 2, this one and this one. These 2 can get separated like this. The same way you can see that they got separated. If the separation is like this is called as alternate, and this is called as the adjacent. This is adjacent 1. Adjacent 1 is this has gone this side, this has come this side. Adjacent 2 is this side has come this side, this has come this side. So these are the way the poles are there. Now the alternate segregation, the first one. Here, what kind of gametes will be there? In one gamete, you will have this red and this. This is there. In another, you will have this and this. Each have the 2 chromatids. So you have this red one has gone here, this blue one has there. The other one will have the other red chromatid, other blue chromatid. So these 2 are the normal type. The other 2 is having this part of red and blue, one chromatid of this. So this is the translocated chromosome. In these 2, translocation is not there, in these 2, translocations are there. So this is one gamete, this is gamete 1, this is gamete 2, this is gamete 3, this is gamete 4. All these gametes are having complete genome, because you can see this is full red chromosome, full blue chromosome, the same thing here. And this is having this part of blue here, rest of the blue is here. This part of red here, the rest part of red is here. Again the same thing. That means in all of them, they have the complete genome, hence each one will be viable. They will be viable. They will not die. The adjacent one, let us see what will happen. In this, the red one, this is normal, this red one is normal. The blue one with red, this is there. So this is one chromatid of this, another chromatid of this, but these two are not having the complete genome. I'll go back and I'll show you once again. So these two are not having the complete genome. Same thing is the case here, that if this and this is together, they are not having the complete genome. I am going back and I am again showing you that these two have the complete genome. These two, if I say this part of the red, so when it is the adjacent one, in this case, this part has come up, this part has come down. So here, this red and this red, that means red is more, blue is less. Blue is not having the complete genome. Red part is being duplicated. If you see A, B, C, D and so on, you will find that there is duplication. Same thing is here, this blue is complete. This blue part is the duplication of this red part, and its next red part is not there. So this is also incomplete, this is also incomplete. You can see again back here that all these are having the incomplete genome. So they are none of the gametes thus formed is having complete genome, hence they are non-viable. Let us go to the adjacent 2 segregation, where the segregation has occurred like this. You can see this is red one, this red one is here complete, but what is this red and this part of the blue is here. Rest part of the blue is not here, that means this is having duplication and this is having a part deleted. So this gamete is having incomplete genome. Here also the same thing, that means this is having incomplete genome. Here also you can see blue more, red less, that means this is incomplete genome, this is incomplete genome. So all these gametes will die because they are not complete. So if I take all the 3 together, you will find that only this combination is having the complete genome, and rest all are not having complete genome, and these gametes will die. So in other words, this will also not give you any viable gamete where the combination is different from the parental. And this is very important. Once again, I will say this is the beauty of the nature, that nature did not allow the recombinants to survive. If the recombinants, if these gametes would have survived, then you would have got newer combinations and a real fatal kind of a situation, that there will be lots of gene missing, lots of gene duplicated, and very complicated condition. So with this, I come to the discussion point that once again I will suggest you draw pairing of the normal homologues with the translocated homologue. You practice this, because as such when I am showing with chromosome, it is not becoming always very clear. If it was chalk and board and had one drawn, one would have seen that how the things are different or not. So, you should draw and try to separate the chromosomes as it happens in anaphase 1 of meiosis at least 5 times. Number 2, whether crossing over occurs only at one place when the two homologues, each of the two chromosomes pair. So, these things you have to discuss. Once again I little bit go back to the one. Here we are not showing any crossing over. Let us say if a crossing over had happened here, no problem. This part would have gone with this part would have gone with this. So, no change would have happened. Same thing, if there is any crossing over at any point, so this chromosome would have gone with this and this chromosome would have gone with this. So, crossing over at any place will not make a combination difficult, the combination will remain the same. That is why the crossing over sign has not been shown. So, with this I come to the end. Thank you.